Hello everybody and welcome to today's webinar hosted by the Centre of Excellence in Severe Asthma. The webinar today will be on COVID-19 recommendations in severe asthma and looking towards treatment and prevention. My name is Dr. Vanessa Clark and I'll be your chair for this webinar. Before I introduce our two fantastic speakers, I'll just go over a few housekeeping rules. Firstly, I'd like to let everybody know that this webinar is being recorded and will be available on our website at a later stage. Secondly, we have two presentations today and you'll be given the opportunity after each presentation to ask questions. To do that, you can type your question into the Q&A box down at the bottom of your screen and we'll do our best to answer them for you. So without further hesitation, it is my pleasure to introduce Professor Peter Gibson. Professor Gibson is a world-leading respiratory physician and researcher. Professor Gibson works at the clinical interface as well as conducting world-leading research into improving regimes and management strategies for airway diseases and asthma. Today, Peter will be introducing the recommendations for COVID-19 in severe asthma. Thank you, Peter.
Thanks, Vanessa. It's a great pleasure to present these recommendations. So the Centre for Research Excellence in Severe Asthma has been working over the last 6 weeks to develop clinical recommendations for COVID in severe asthma. These recommendations are available on the web link you can see at the bottom of the screen, and they're presented as a downloadable PDF and also as a series of infographics. And here we're really trying to capture some of the issues around COVID-19 and in particular the impact it's having on people and healthcare workers around the country and people with severe asthma and those healthcare workers who manage people with severe asthma. The recommendations start by reviewing the impact of COVID-19, and although asthma itself doesn't appear to be a particular risk factor for this infection, the impact on people with asthma and severe asthma has actually been quite profound. And it's in areas of what do you do for normal treatment? What about monoclonal antibody therapy? How do you continue to receive that in a situation of lockdown? Are my usual drugs and devices safe? And how do I maintain well-being during this period? So these are the topics that are covered in the recommendations, as well as a useful set of references and resources for people that are looking for more information about some of these issues. I'm going to go through some of these recommendations now. COVID-19 is the name of the illness that's caused by the SARS-CoV-2 virus. Fever is a very common symptom, as is cough, and curiously altered sense of smell. Most people experience a mild disease from which they recover. However, there's a small proportion that get progressive respiratory impairment, some of whom require hospitalisation, intensive care stay, and this can result in death. The recommendations for asthma management need to be reviewed in the light of COVID-19. So here we see two infographics dealing with these issues. Firstly, it's expected that people with asthma will continue to experience asthma attacks. And essentially, the key recommendations here are that it's business as usual, meaning people need to review and follow their written asthma action plan and use the existing advice that tells them when to increase treatment and when to seek emergency help. Something new in the COVID era is around isolation. What does that mean for people with severe asthma? So although people with asthma don't appear to be at greater risk of infection, because they have a compromised respiratory system, the consequences of an infection could be profound. And so because of that, we're recommending that people with asthma self-isolate and practice physical distancing including from household occupants. During the period of community isolation, we're also recommending that people change their healthcare delivery from face-to-face assessments to telemedicine appointments. What about someone who's working? So if you have severe asthma, our recommendation is that you work from home. Now, some patients with severe asthma may also be classified as essential workers. And our recommendation here also is that these people work from home. We anticipate this recommendation will change as the social distancing requirements change in Australia. But right now, we're recommending social distancing, staying at home and working from home. What about asthma drugs and devices? So the influenza season is approaching us now, and it's important that people with severe asthma have influenza vaccination to protect them from influenza. This won't protect people from COVID. However, there is an unknown about the risk of dual infection. What happens if you got an influenza virus? Would you be more susceptible to getting another virus? So it's a double reason to be more vigilant and have influenza vaccination. Delivery of asthma medication by puffers continues to be recommended and safe. It's important that people don't share their puffers. However, we recommend against nebulizer treatment. There's a range of devices, nebulizers, high flow oxygen, positive pressure ventilation, that generate aerosols. In generating aerosols, there's an increased risk of viral spread. So for this reason, we recommend against these procedures. Spirometry is the subject of careful review by the Thoracic Society, and the current recommendation is to perform this under usual infection control measures. Inhaled corticosteroids are the key asthma preventer, and they should be continued as usual. Oral corticosteroids are needed as rescue for severe attacks. They can continue to be used as rescue for severe attacks. Now, an important issue is monoclonal antibody therapy. So this is a key treatment for some people with severe asthma. And in our document, we present both an infographic and text-based information about how to access and administer monoclonal antibody therapy during this time of the COVID pandemic. If we look at the infographic, the key issue here is that the model of care needs to change during the COVID pandemic. Most severe asthma clinics are located in hospitals, which will also be expected to treat patients with COVID-19 infection. Therefore, to minimise exposure, we recommend transferring administration of monoclonal antibodies to the community. Fortunately, there are several options to make this possible. People could receive their monoclonal antibody from their GP. Alternatively, the nurse administration programs could be used for either Nucala or Fasenra. Or people could self-inject for either Xolair, Nucala or Fasenra. So this means there'll need to be an active planning between the patient, the severe asthma clinic healthcare professionals and the community services to identify what's the best way to transfer administration of monoclonal antibodies from the severe asthma clinic to the community. At the bottom of the infographic is an example of how we go about this at our hospital, the John Hunter Hospital. And importantly, it's recognised that social isolation and the fear of the pandemic can have a very profound effect on your well-being. There are clear examples of what happens to stress and anxiety during pandemics. There's a high risk of community concern, specific concerns around the illness, and people exhibit a range of behaviours in response to this. They seek a lot of information to be reassured and feel in control, and other people exhibit behaviours that are not considered helpful for their well-being. This means that we need to actively maintain well-being during this time. And there are some important suggestions in the document about how this can be done. The key areas are maintaining social connectedness, controlling media intake, maintaining nutrition and physical activity. And this information not only applies to people with severe asthma, it's also important for clinicians. As clinicians, we are responsible for maintaining our own well-being so that we can be there to help people manage their illnesses. So these are the clinical recommendations that the Centre for Severe Asthma has put together to help people and health professionals manage their illness at this time. I'd like to thank a number of people who contributed to this. Firstly, the writing team. Secondly, the critical review that was provided by chief investigators in the Centre for Severe Asthma. And then lastly, with the excellent visuals from Olivia McDonald and Jane Green. So just before ending, I've told you about our clinical recommendations. I want to pause for a minute and think about asthma and coronavirus infection risk. What does the science tell us about this? I think we were all expecting asthma to be a major risk factor in COVID-19, and we were surprised when it didn't turn out that way. Asthma was underrepresented in patients hospitalised with COVID-19. Here's an example of two studies. This is a large study from the UK involving over 16,000 people hospitalised for COVID-19. Their mean age was 72. 14% had asthma, which is roughly, it's within the range of the normal prevalence of asthma. So there wasn't an overrepresentation of people with asthma being hospitalised. And although these people had many comorbidities, asthma was not associated with in-hospital mortality. The forest plot on the bottom shows that age was the biggest risk factor for in-hospital mortality, followed by chronic diseases such as cardiac disease, COPD, and other chronic illnesses. Secondly, when we look at China, in one of the early reports of 140 patients hospitalised with COVID-19 in Wuhan, none of the patients reported comorbidity with asthma. So here we have representative evidence that although asthma is a concern, it's not showing up in people hospitalised with COVID-19. Why might that be the case? A feature of COVID is that blood lymphocytes and eosinophils are suppressed. They're the cells that drive inflammation in asthma. Maybe that's the reason. Alternatively, it could be related to the receptor for the coronavirus. This is an epithelial receptor called ACE2. It's used for viral entry into the cell. And it's been found that when you look at nasal epithelial expression of ACE2, it's actually reduced in allergic individuals and people with asthma. So if you look at these two graphs and compare the black box plot to the red one, people in the red one have asthma and allergic sensitization, and they have increased expression, they have reduced expression of the ACE2 receptor. The adjacent graph shows that the more intense the allergic sensitization, the greater the reduction in expression of the viral receptor. There's further work showing that if you challenge adults with asthma this further lowers ACE2 gene expression. So on the one hand, we're seeing that the type 2 response, which is typical of allergy and allergic asthma, reduces the viral receptor. You can then ask the question, okay, what happens if someone gets a viral infection, such as a common cold? And here we see what happens to the ACE2 receptor when nasal epithelial cells are infected with rhinovirus. And you can see in the orange box plot, there's a dramatic increase in the ACE2 receptor infection. So whilst we have evidence that T2 inflammation reduces ACE2 gene expression, and this may explain the apparent lack of asthma being prevalent in hospitalised cases, we have this second issue that respiratory virus infections increase ACE2 levels. And it raises the question, okay, what will happen if a person with asthma got a coexisting virus infection? I think what this tells me is we need to continually be vigilant about these issues, and with the relaxing of social distancing and the impending influenza season, we need to bear this in mind and be particularly cautious in our efforts at maintaining infection prevention and also monitoring and treating exacerbations with asthma. Thank you for your attention, and I hope you find these clinical recommendations of use.
Thank you for that, Peter. That was a great presentation. We'll now invite some questions. So the first question is, should the recommendations for isolation of patients with severe asthma extend to their household as well, as long as it's practical?
Yeah, that's a good question, and we've said yes to that. So we've said the person with asthma, with severe asthma, should look at isolating from other members of the household where that's practical.
Okay, the next question I have here, do the findings about asthma and COVID-19 apply to severe asthma or asthma only? That is, if you looked at someone with severe asthma patients, would they like a greater risk of negative outcomes compared to mild or moderate asthma? And is there also a behavioural component? So people with asthma or severe asthma are more concerned about the exposure and take fewer risks around exposing to that exposure than people without disease.
Yeah, so the issue, we haven't found any evidence specifically addressing severe asthma. It hasn't shown up in any of the large series, so we can only assume that they would have contained some people with severe asthma and they haven't presented as having an increased risk. So all I can say is at this point, we don't have evidence that severe asthma itself presents a risk of infection. But like other chronic illnesses, the host or the respiratory system is compromised. So if that person got any infection, they would have an increased illness burden because of their underlying disease severity.
Okay, somebody asked on the chat function about the mechanism behind the altered sense of smell, and I believe Nathan answered that coronaviruses are known to infect the CNS, possible that the nasal infection leads to infection of sensory nerves in the nose. And then there's another question here. If COPD is a comorbidity secondary to cardiac, then the genes which are expressed in COPD, which are not in asthma, may be key. So that might be more of a comment.
Yeah, I think it's clear that there's differential disease susceptibility, and that's going to throw up important research questions that will, I think, need to be answered as we move forward with this.
Any other questions from the floor? Okay, well.
So I'm going to stop sharing my screen, Vanessa.
Okay, that'd be excellent. No other questions that I can see coming in. All right, we might move to our next presentation and perhaps people might be able to think of some questions at the end if there's any more. Okay, so it's now my pleasure to introduce Associate Professor Nathan Bartlett. Associate Professor Bartlett is the head of the Viral Immunology and Respiratory Disease Group based at the Hunter Medical Research Institute. Associate Professor Bartlett is currently exploring novel therapies and treatments to fight COVID-19. His team are exploring a range of antiviral options for this virus, partnering with other researchers to explore different methods to fight the virus. Today, Nathan will be speaking on I Want My Life Back, looking towards treatment and prevention of COVID-19. Before we hear from Nathan, he would first like to play a segment of a radio interview, which he recently recorded with Kia Handley for the ABC radio.
The big V word, it feels like a lot is riding on that in your life at the moment. You know, people are working on it, but you're hearing it could be 12 months, 18 months away. But is there another option? Is a COVID-19 vaccine the only medical way to fight this virus? The short answer is no. For the long answer, I found a Hunter Medical Research Institute researcher to talk you through that. Associate Professor Nathan Bartlett, good morning.
Good morning, Kaya. How are you doing?
Good, thanks. Hey, tell me, firstly, why do we hear so much around vaccines when it comes to something like COVID-19?
Well, over the long term, vaccines are going to be what provides broad community-based protection. And so they're extremely important for the long-term strategy to control and essentially live with this virus. But that's really the long-term game. In the short term, we're still going to be seeing infections occur, and obviously they need to be treated.
As this started to happen, this global pandemic, how quickly did you realise that you needed to switch your attention to focus on this?
Oh, I've been interested in respiratory viruses for most of my career, and I've understood that they pose an ongoing, major problem for human health. It was only a matter of time before something like SARS-CoV-2 emerged and caused the pandemic we're now seeing. So, I've been aware of this medical research need for quite a while. So we had a lot of the tools already in place so that we could very quickly pivot to focus on studying SARS-CoV-2 and development of treatment approaches.
So you're looking more at an antiviral treatment. What does that mean for someone like me who's not medical?
It can mean lots of things, and it's really there are multiple approaches that could be used in addition to a vaccine that would provide both protection as well as treat active infections. And so, the classical definition of an antiviral is a drug that directly inhibits the capacity of the virus to replicate. So they normally inhibit or interfere with the processes the virus needs to access in order to reproduce itself and spread. And that's a classic antiviral. These are usually small drugs, inhibitors that block key viral proteins that the virus uses to reproduce itself. So there's certainly one approach that's being investigated intensely at the moment. But there are also other ways you can interfere with the virus's capacity to replicate. You can modify the capacity of the host, of us, to respond to the virus. So we can do that by boosting our immune system. And we're looking at approaches that do that, but we can also look at the cellular processes that the virus needs. The viruses are intracellular parasites, and they need host or our cell processes in order to replicate. And so if we understand what cellular processes the viruses use, and we can modify those cellular processes, we can also interfere and potentially block the capacity of the virus to replicate and spread.
Yeah, when you get a new virus like this, is it like starting from scratch? You know that these base things work, but you don't quite know how it's going to respond with this virus?
That's, we have some ideas, and we can take guidance from other related viruses, particularly around the cellular processes, because a lot of these cellular processes are the same and used by multiple different types of viruses. So we can automatically begin to focus our attention on some of these understood cellular processes and use drugs that are available that have been shown to be effective against other viruses that target these cellular processes. So we do have a very substantial wealth of knowledge around these that we can then begin to start testing specifically against SARS-CoV-2.
How do you even test that when, let's say, in the early days, even now, you know, there's still a lot we don't know about the virus. There's maybe not access to samples. How does that all work from your perspective, Nathan?
Well, there are models we can use. So we can use related coronaviruses. All coronaviruses use the same strategy to replicate. And so we can use some of these less pathogenic coronaviruses, which circulate every year through the community. And we can test these approaches against those coronaviruses and have a really good sense that they would work against SARS-CoV-2, which is just a variant really on these commonly circulating coronaviruses, and essentially it's very similar in the way it replicates.
So how's it going? What are you up to? What can you tell us?
Well, we're testing a number of approaches using our various less pathogenic coronaviruses, and we're starting to see some interesting results. I can't really divulge much more than that at the moment.
No, I thought that might be the case.
yeah, but certainly we are trying a lot of different approaches, and you know, we're reasonably optimistic that one, if not more, will show signs of being effective, and thereby be able to move them forward to test against SARS-CoV-2 directly. Yeah, and
I imagine that you're not the only one doing this as well. How are people communicating around the world as everyone's trying to answer the same question?
Yeah, I mean, I think that's been one of the success stories, if you want to look for a silver lining in this pandemic, has been the way that the medical research community has banded together. Even the politicians have actually shown some unity as well in helping us through this. But it's been especially evident with medical research and very open sharing of data. I mean, we had the sequence, the genetic sequence for SARS-CoV-2 uploaded and available for all researchers very quickly after it was isolated in China. And that's really been indicative of the collegial and collaborative spirit with which the global research community has embraced tackling this viral challenge.
It's 10 minutes away from 10 o'clock. You're on ABC Newcastle Mornings. You're hearing from Associate Professor Nathan Bartlett from the Hunter Medical Research Institute. We're talking a little bit about other treatments beyond a vaccine to COVID-19. Now, we have been hearing about other drugs, mostly out of the US, that may or may not work. You're doing a little bit of work around this with cancer drugs. Tell us about that, Nathan.
Yeah, so there's been a large focus on what we call repurposed drugs. So these are drugs that have already been approved for clinical use for other indications, other diseases. So they've been through the regulatory processes, approval processes that are quite involved and take a long time. So this, using repurposed drugs, enables us to very quickly move a drug into a clinical trial for COVID-19. And so we're looking at other diseases with such drugs that are approved. And one area we've looked at are cancer drugs, and particularly drugs in that space that target growth factor receptors. It's easy to imagine that growth factor receptors would be involved in a disease like cancer where cells grow uncontrollably. But viruses also hijack growth factor receptors in a number of ways to help infect and replicate and spread. And so it is quite sensible to speculate that these growth factor receptor inhibiting drugs that have been approved for cancer might also be effective in the context of COVID-19.
And we know that speeds up some of the process, right? If the drug is already out there, already being used, already being tested, that is a good thing for like the whole process.
Correct. I mean, the big time-consuming component of drug development is really safety and determining that a drug is safe for use in the population. And so this takes a long time. So if a drug has already been through that safety testing and approval process, then that massively speeds up the capacity to get it into a clinical trial.
As a region, as a state, we're sort of counting down the days till Friday when we see some of those restrictions being lifted around New South Wales. But we are still going to have to live in the community with this virus. How have you been working with the Hunter community to make sure that we're in the best position to do that?
Yeah, so on one side, obviously, we have this medical treatment vaccine area of research, but day to day, looking at other approaches that are going to make living with this virus, and that's something we're going to have to come to terms with. We're going to have to live with this virus for the foreseeable future. And so things we can implement in our day-to-day lives that make that safer. And so we're working with local businesses that have changed their business models to look at development of sanitizers that can be used for hands or for surfaces, materials that you can coat surfaces that are regularly touched by the public, touch screens in various scenarios that have intrinsic antiviral activity. So multiple approaches that you can implement into everyday life that are all going to restrict the capacity of the virus to remain infectious and thereby cause infection in community.
Have people been open to that in the community? They want to do what they can?
Absolutely. I've been inundated is the only way to describe it, with requests to help various businesses test materials, sanitizers, etc., that they think will be useful for community implementation.
And so you're looking at that to see if they are actually effective, like other store-bought stuff?
If it's effective, sort of alcohol-free sanitizers. So we had a big issue with alcohol supply at one point, provides 70% alcohol for disinfectant. So the supply chain for that can be quite unpredictable. So alcohol-free sanitizer options are a real focus at the moment as well. And plus that, alcohol on hands over extended use can be quite damaging too. So we are looking at other approaches that could be implemented into everyday life that people would be happy to use on a regular basis and would be, safe and not irritating.
As you said, Nathan, as a virologist, this is something that you think about quite often, this sort of hypothetical global pandemic. Now that you're living through it, working through it, is it what you expected?
Is it what I expected? It's hard to answer. Yeah, I guess. I suppose. I mean, I guess we're fortunate to live in a country like Australia where we've been able to implement, some of the social distancing and other lockdown-related rules that have been very effective in containing spreads. So I think for us, it's been a very different experience to say what it's currently like in the US or Britain or Europe. So I'd be very interesting to talk to my colleagues abroad over the next few months to share our, relative experiences in this.
Yeah. Nathan, always great to catch up. Thank you. Best of luck with the work that you're doing.
Thanks, Kai. Great to talk.
Associate Professor Nathan Bartlett there. He's a virologist with the Hunter Medical Research Institute, talking you through some of the other options that are being looked at when it comes to treatment, prevention of COVID-19.
Okay, that was great. And now we're going to hear from Nathan. Thank you, Nathan.
If you just heard, really to drill down on some of the treatment approaches and really why they're going to be necessary if we're, ever going to get our life back. I've had a number of comments from people around, well, Australia hasn't really been affected. We've got no one really sick with COVID-19. Why do we need to worry about a vaccine or a treatment? Well, I think, you heard very nicely from Peter in the previous presentation that the anxiety and the risk and the threat is still there, and it's having a huge impact on quality of life. And, that because we haven't succumbed to the first wave doesn't necessarily mean we're going to not be affected as winter comes on and there's a potential for a second wave. And going forward, we're going to need to have treatment options and ultimately a vaccine if we are really going to be able to get back to any semblance of our previous life. Okay, so I just thought I'd give an overview on the treatment approaches and how they really fit into the spectrum of infection and disease around COVID-19 and what to target. And really when we think about this, we consider the virus. So we consider treatments that might directly target the virus and prevent the virus from getting into cells and replicating. And so the type of treatments there might affect the ability of the virus to replicate its genome or RNA replication, because these are RNA viruses, or the proteases the virus encodes that it needs to process proteins that it needs to assemble itself to make more copies of the virus and then to move out and infect other cells. So we can think about approaches that target the virus directly, but we can also think about the host. You and me are the host for this virus, and we can think of treatments that are going to improve our ability to either prevent infection, so preventative approaches, but also treatments that are going to improve our ability to clear the virus, reduce the capacity of the virus to replicate in our cells, in our airways, and thereby restrict the ability of the virus to cause disease. And these approaches, there's a spectrum of approaches that sort of fall into this framework, and that all ultimately impacts on disease outcomes. That's what we're aiming to achieve, reducing those disease outcomes. Just to say, different approaches have different timeframes typically. You heard me talk about repurposed drugs in the interview before. So these drugs have already been through the approval processes and are likely to be much quicker to get into the clinic. A vaccine, however, is an entirely new drug or a new treatment, a new medicine, because it's against a new virus. And these can take typically multiple years to develop. That's assuming we can develop an effective vaccine for SARS-CoV-2. Just remembering that we've never developed a vaccine for this type of virus before, so there's no precedent for it. So we're, optimistically looking to develop one, but there's certainly no guarantee that we will. But even if we do, it's no doubt that it's going to take a number of years before we have one that can be safely administered to the community, and thereby there's still going to be some time that we are still entirely susceptible to infection and will need treatments. But even when a vaccine is implemented, we are still going to need treatments because people will still become infected. So there have been a lot of focus on antivirals that have been developed for other viruses. I mentioned, we can take a lead from our understanding of how other viruses replicate and ask, are drugs that are effective against other viruses, are they effective against SARS-CoV-2? One virus that a lot of drug development attention has been focused on is HIV over the last 20, 30, 40 years. I guess now since the 80s, obviously HIV was a huge, was a pandemic which caused millions of infections and deaths. And so there's been a wealth of research into HIV, and as a result, many drugs have been developed for HIV. And so there's been some interest in understanding, will these drugs be useful against SARS-CoV-2? I've listed a few of them here. Remdesivir, which I'll talk about below, and some other ones that affect other functions of the virus HIV, affect protease or affect reverse transcription. I'll talk about two of them. The rationale for remdesivir is quite sound. HIV is an RNA virus. When I say RNA virus, I mean that the genome, the genetic code that defines the virus is made of RNA, both HIV and for coronaviruses like SARS-CoV-2. So they have a similar type of genetic structure. So they use RNA to make their genome. And so things that interfere with the capacity for them to replicate their RNA genome may be effective across the board. And certainly remdesivir is one such drug, which looks like it may have some efficacy. So as I said, it can target multiple viruses. It is authorized for it is now authorized for emergency use in the USA. It's being tested in Japan. There is some data coming out that it is modestly effective. It's not a huge clinical benefit, but there does seem to be some efficacy. But that needs to be balanced somewhat with some side effects. So this drug does have some side effects as well. Other approaches we can think about are targeting the structural components of the virus particles, such as its protein capsid, or the proteins that stick out of the virus particles, such as the spike protein that mediates binding of the virus to cells. And we can look at drugs that bind to these and inhibit the capacity of the virus to bind to cells and thereby get into cells and initiate infection. So again, these tend to be more virus specific because coronavirus spike proteins are very structurally unique to coronaviruses. Even the spike protein of SARS-CoV-2 is quite unique to SARS-CoV-2. And that's why you need an immune response specific to this spike protein, and immunity to other coronaviruses is not protective due to those structural differences in the spike protein. Varying efficacy due to the capsid protein variability. Again, these proteins, as I said, tend to be quite variable. It helps the virus evade immune responses, but it does provide the potential for the development of drugs that act a bit like neutralizing antibodies that bind to these surface proteins and prevent the virus from binding and entering cells. Another HIV drug, which I listed on the previous slide, is a combination medication called leponavir/ritonavir. It's a protease inhibitor, and this is one that I find a little less obvious for having an indication for coronavirus, because it targets an HIV protease, which is very different to anything SARS-CoV-2 makes. So the fact that this would be considered to be effective against SARS-CoV-2 when it's inhibiting a protease from a very different virus is a little less obvious to me. But nonetheless, there was some evidence in vitro that this drug combination was blocking SARS-CoV-2 replication. However, recent data from a randomized control trial with this drug showed absolutely no benefits with treatments. So it was compared to the standard care alone, which is supportive treatment, and there really was no difference in terms of the disease development, mortality, and in addition, treatment with this HIV medication also had some fairly notable adverse events or side effects. So not only were you just as sick, but you also had the added side effects or adverse events associated with treatment with the drug. So all in all, in a clinical trial, this really did not seem to be particularly useful. I guess just highlights a word of caution that, you know, in our rush to get approved drugs with some evidence, and often that evidence is quite limited, evidence of efficacy against SARS-CoV-2, and often it is only in vitro in a tissue culture dish. You stick a drug on infected cells in a tissue culture dish and you kill the virus-infected cells. That is no way predictive of it being effective in a clinical trial. And I think we're seeing quite a lot of that at the moment, that these very limited preclinical evidence is really not providing good support when we see no efficacy in the clinical trials. So there's some of the antivirals that are currently being tested. Other approaches we can look at, as I mentioned, affect the ability of the virus to bind to the cell's virus receptor, and Peter very nicely introduced the receptor for SARS-CoV-2. ACE2 is the human cell protein that the virus binds to in order to gain entry into cells. It also needs another host protein, an enzyme, a protease, which processes the spike protein, enables the spike protein to efficiently bind to ACE2 and enter the cells. So there are a couple of human proteins on the surface of our cells that are important, that are used by the virus to get into cells, and these are potential drug targets. Okay, so there are drugs in development that look to block ACE2 or inhibit that activating protease, as I just mentioned. Okay, and so these are in development, and these, you know, certainly the ACE2 blockade drugs would act a lot like neutralizing antibodies, which again might block the capacity of the spike protein to bind to that ACE2 receptor. We mentioned other cellular processes that are used quite commonly by multiple virus groups, and one are growth factor receptors. You heard me mention those again in the previous interview. There's a lot of drugs that have been developed in the cancer space for these growth factor receptors, so there are a lot of therapeutics available So it's a rich source of potential approved drugs that might also be effective for SARS-CoV-2. And, you know, so there's a lot of rationale for this. The growth factor receptors are used for viruses to help bind to the surface of cells. These growth factor receptors and the signaling that they undertake also induces cellular processes that the virus is able to hijack to facilitate replication. So we're also interested in looking at drugs that target these growth factor receptors. And it is also thought that a drug I'm sure a lot of you have already heard about a lot, hydroxychloroquine, it is thought that it also impacts growth factor receptor activity through affecting processes of virus uptake. One class of drugs that I'm particularly interested in, because it does have very strong clinical data, are macrolide antibiotics, such as azithromycin. Now, I'm sure many of you would have heard of azithromycin. It's a very common antibiotic used for bacterial infections, but it actually turns out that this is a very interesting class of therapeutics. It has many effects beyond its antibacterial activities. We've noted that it can affect the immune system, so it has immunomodulatory effects, or be anti-inflammatory, suppressing inflammation. And we also identified some antiviral activity with macrolide antibiotics. So certainly the anti-inflammatory and the antiviral activity is something we'd be very interested in, as it would make sense for it as a therapeutic for SARS-CoV-2, where we know inflammation, and certainly antivirals are going to be therapeutically useful. The reason we're particularly interested in azithromycin is it does have very compelling clinical data to support efficacy in a disease like COVID-19. So this is work from Peter Gibson, published a couple of years ago in The Lancet, which looked at azithromycin treatment in patients with asthma. And it found very convincingly that patients treated with azithromycin, I think over the course of a year, had significantly less numbers of asthma attacks or exacerbations over the course of that year. So if we look at this graph here, the azithromycin treated group showing clearly a lower rate of asthma exacerbation compared to those randomized to the placebo group. Okay, now we know that a large percentage of asthma exacerbations are caused by respiratory viruses. So this leads us to wonder whether or not part of the efficacy of azithromycin in this context is through its capacity to be antiviral or suppress virus-induced inflammation, and then part of the effect we're seeing is related to that, which then provides good rationale for asking, will it be effective in the context of COVID-19? So this is certainly something we're very interested to test and have initiated experiments to do so. Back to our old friend hydroxychloroquine. This has been Donald Trump's favorite drug. And so as a result, it's obtained substantial media attention. And, you know, it does have some things going for it. Certainly it's inexpensive, easy to make, widely available. It's approved. It's understood in terms of its safety and dosing, etc. So, you know, it has a lot of positives around it. However, there's been some mixed results. So it's been essentially the drug of choice for a lot of these early COVID-19 clinical trials. And so we're starting to get some data now around its efficacy in the context of COVID-19. One of the earliest clinical trial data to come out was one from a French clinical trial, which was published a couple of weeks ago. The reference is down there. And it suggests, it's a very, very small trial. So there are 20 patients, which in the context of a clinical trial is essentially meaningless. But given the urgency of this, of the situation, that they published the data from a very small cohort, and they reported that they saw reduced viral load 6 days after treatment. So it did appear that those patients, given the macrolide, given hydroxychloroquine alone, and actually azithromycin even improved that, seem to do better in terms of clearing the virus more quickly. Since then, however, a more substantial clinical trial, data from a more substantial clinical trial has been published. So we're talking not 20 patients, but 14,000 patients. So slightly more statistically robust data. And they looked at two main outcomes. One was deaths and one was cardiac arrhythmias. I'll show you the mortality data first and really just draw your attention to the hydroxychloroquine or chloroquine treated outcomes down here. And you can see invariably that those patients who were given chloroquine or hydroxychloroquine, these are two related drugs, did worse. They had a higher risk of death than those given standard care. So with this much larger clinical trial, it seemed that actually hydroxychloroquine or related drugs were actually a lot worse for you than the standard of care, suggesting that probably they're not a good idea in this context. And it's thought that this is certainly related to the fact that these drugs have cardiac complications. And if you look at the data here, which is around ventricular arrhythmias, so this is arrhythmias, heart arrhythmia, you can see again that there was a much higher prevalence of heart arrhythmias in the patients given these medications as well. So certainly thought that a lot of the morbidity may have been around, morbidity and mortality may have been around these heart complications, which are clearly associated with treatment with these drugs. So all in all, I think the data for hydroxychloroquine now is that it's probably not going to be effective for COVID-19, and if anything, it's probably going to result in worse clinical outcomes. So I'm going to shift gears very quickly now from antivirals directly to talk about other approaches. One thing we're interested in looking at is being able to boost immunity. We've heard a lot about adaptive immunity in the context of vaccines. This is really another approach which looks at the innate immune system. The advantage of the innate immune system is it's not going to be specific to one virus. The innate immune system is an early immune response system that is not going to be reliant on recognizing a specific virus. So we can recognize shared viral proteins or markers that'll activate innate immunity generally. And so we're working with a company that's developed a TLR2 agonist, and this is really what we're hoping will bridge the gap between a vaccine and antiviral. It'll provide through inhaled or a nasal spray type delivery, provides enhanced immune protection in the airways, make you more resistant to infection and less likely to get more of those severe disease outcomes that we know a subset of patients who are infected with SARS-CoV-2 end up with. And the other thing I just wanted to touch on before I finish up is it's one thing to have a good drug which seems effective in vitro, in tissue culture in the lab, but a lot of the issues with these drugs is there's no way to get them to where they need to be, and that's in the airway. So hydroxychloroquine and these HIV drugs, these are typically oral drugs, so there's systemic exposure to these drugs and a lot of side effects, off-target effects, because the drug is not being delivered directly to the infected airway cells. Has to travel through the circulation, and it can affect multiple tissues and organs, and there are a lot of side effects associated with that. So what we really need to also think about is effective ways of getting these drugs directly and safely to where they need to be, that is the airway epithelial cells that are in which the virus is replicating. So we're doing a lot of work optimizing what we call nanoparticles. These are delivery systems that we can wrap up a drug in. They can be inhaled, and this will chaperone the drug directly into the airway epithelial cells, where it can then act on the virus directly and shielding it from other cells and tissues where it might have adverse toxic effects. So not only do we need good drugs that are efficacious, but we need effective ways to get those drugs to where they need to be. And certainly there's a lot of interest in nanoparticles as a way of doing that. And I'll finish now with a quick chat about vaccination. It's certainly not a new approach, and there is a reasonably successful track record, although there are a lot of viruses which we still haven't got vaccines for, and a lot of those are respiratory viruses. But the goal is to induce sustained memory. So this is protective immunity against the pathogen of choice. And how does it do that? Well, you do that by showing your immune system something which looks like the virus. You generate antibodies, and the B cells that make those antibodies are long-lived. So you get production of these antibodies over prolonged duration, and these antibodies will provide protection. But as I said, vaccines for RNA viruses are challenging. You have to remember, once you set out to make a vaccine, what you start with is what you have after 2 years. So you basically, you've made your bets early, and you hope that the vaccine you end up with 2 years down the track is still effective against the virus that is now circulating at that time. And that might not always be the case. And we certainly know that with influenza, we need to update our vaccine for influenza on a yearly basis because the virus does change. We don't think that'll be the case for coronavirus, but there's certainly no guarantee that the virus will be stable over that duration of time. And I think I will, in the interest of time, I will just skip to my summary slide. And just to overview everything we've discussed, we've talked about direct antivirals, we've talked about indirect antivirals, so helping your cells inhibit the infection, and there's a list of things there. And then I didn't talk too much about passive immunity, but certainly active immunity with vaccines. And really the aim ultimately is to bring the group hug back, and one day hopefully we'll be in a situation where we have treatments, we have a vaccine that'll enable us to safely enjoy each other's company again. So thank you very much.
That was wonderful. Thank you so much, Nathan. I'll ask for some questions from the audience now. We do have one question waiting here, which is closer, a vaccine or a treatment?
Closer. Look, certainly there's been a lot more work put into vaccines, but I think in terms of how quickly we'll get something to the public, I would say a treatment's probably going to be easier to get out to the public. Vaccines are just very hard to develop. To prove a vaccine is safe and effective is incredibly arduous. You have to test it in literally thousands of people, wait for them to get infected, and then try and determine whether or not there's protection. So I would say that a treatment is more likely to get approved and be available quicker than a vaccine is.
Okay, thanks so much for that, Nathan. I've got another question here. What is your opinion on the use of ACE inhibitors? ACE inhibitors.
It depends on how the inhibitor works. So angiotensin converting enzyme 2 is an enzyme involved in converting angiotensin, obviously. If that inhibitor is also going to block the capacity of the virus to bind, then certainly it might work. It all comes down to understanding the mechanism of action of your ACE2 inhibitor. If your ACE2 inhibitor is infecting an intracellular signaling process downstream of that, external component of the receptor, then maybe that's not going to have any effect on the ability of the virus to block. So again, it just comes down to understanding the fine details of the mechanism of action of that inhibitor to sort of sensibly predict whether it would be useful or not.
Okay, that's fantastic. Are there any further questions? No? Well, given that it is 11:30, we can end this webinar now. I want to thank you for attending on behalf of the CRE in Severe Asthma. And please check out our Severe Asthma website and Severe Asthma Toolkit for this webinar at a later stage. Thank you.